Thank you for the nice introduction. So my name is Patrice Mimche, and I'm an assistant professor in the Department of Pathology. So my lab is focused on elucidating the role of neuronal guidance cue in the pathogenesis of inflammation and fibrosis. So fibrosis is an exaggerated wound healing response subsequent to chronic tissue injury that will cause the destruction of organ architecture. And so this is due to the highly deposition of collagen inside the tissue. And this will lead to the dysfunction of the organ. As you can see in this illustration, fibrosis can affect almost various organs in the human body. So in the United States, fibrotic diseases are responsible for up to 45% of death. But fortunately, a therapeutic option for the management of fibrotic disease are severely limited with organ transplantation. In my laboratory, we discovered a pathway that we think, if we block, may potentially reverse fibrosis. In the next couple of slides, I'm planning to share with you my story and how I became a scientist working on fibrosis. So I grew up in Yaoundé, Cameroon. And when I was there, I had very little interest into science. So my main area of interest was to play for the local youth basketball team there in Cameroon. So with the dream of becoming a professional basketball player. So after my graduation from high school, I attended the University of Yaoundé 1 to study biochemistry. So my love and passion for science really started when I was working in a malaria lab in the Pathology Center of the University of Yaoundé 1. Why working on malaria? Simply because it is a very devastating parasitic disease, and it basically is the leading cause of death of children in sub-Saharan Africa. I personally suffer a lot from this particular disease. So after graduating with a master's degree from the University of Yaoundé 1, I was offered a postdoctorate scholarship with the European Malaria Graduate School, and I was stationed at the London School of Hygiene and Tropical Medicine. And my supervisor there were Dr. Livia Vivas and Donatella Taramelli, two leading experts in malaria drug development. So the focus of my doctorate was to evaluate the immunomodulatory properties of novel anti-malaria drugs with the hope that they can be used for the treatment of severe or cerebral complications of that particular disease. So this work basically inspired my next move, which was to work in the immunological basis of cerebral complications of malaria. So after completing my doctorate degree there in England, I moved to Emory University in Georgia, and there I did my postdoctoral training in the laboratory of Dr. Tracy Lamb. And the focus of my work there to investigate the molecular mechanism of neuronal guidance cue in the context of cerebral complication of malaria. So it is when I was working on that particular project that I discovered that the receptor tyrosine kinase that we call BEF-B2 is very important in the pathogenesis of inflammation and fibrosis into the liver. This particular discovery shifted the focus of my research interest from malaria to fibrosis. So in this picture, what we can see here is liver section of mice infected with rodent malaria parasite, where we can see the collagen fiber in these particular sections, and this seems to be reduced in the absence of this particular receptor tyrosine kinase FB2. So I was blessed to be offered a faculty position here by the University of Utah in the Department of Pathology to set up my independent laboratory to work on fibrosis. What we are focusing right now is to investigate the molecular mechanism of FB2 in the context of fibrosis. And FB2 is a receptor tyrosine kinase that binds to the ligand called the Deciphering. So they form the largest class of receptor tyrosine kinase in the human genome. One specificity about this signaling system is that they are involved in various biological systems relevant to fibrosis, like inflammation, cell remodeling, and also cell proliferation. During fibrosis, the main cell type responsible for the production of collagen is a cell called myofibroblast. And we discovered that when a cell that expresses the different ligand bind to a fibroblast that express FB2, so this will lead to activation of the FB2 receptor through a process called phosphorylation, followed by the recruitment of signaling molecule. So this will eventually lead to the transformation of a fibroblast into a myofibroblast producing collagen. We also discovered that blocking the FB2 receptor on the fibroblast using either a genetic approach or a pharmacological inhibitor block entirely this particular process, and it can be used for the development of novel fibrotic drugs. In collaboration with various investigators here at the University of Utah, we are testing this novel idea across various fibrotic disease. And we are now generating some data in the context of skin fibrosis with Dr. Deciphering, and also in the context of lung fibrosis with Dr. Mimche. And what we can clearly see here is that in the absence of FB2, fibrosis, depicted here by the blue staining of collagen, is drastically reduced in the absence of FB2. So this work is currently supported through a grant from the Scleroderma Foundation and the National Institute of Health. In collaboration with Dr. C.M. Bujina, we also started to explore the role of FB2 in the context of fatty liver disease. And fatty liver disease is a pathology that is associated with obesity and type 2 diabetes. And what we discovered is that when you fed mice high-fat diet for them to develop inflammation in the liver and fibrosis, this particular process is drastically blocked in FB2 deficient mice. And we recently received another one to investigate novel pharmacological inhibitor of the Deciphering signaling for the treatment of the fatty liver disease and associated metabolic disorder. With that, I would like to thank all the various members of my lab who are contributed to this work, and also colleagues here at the University of Utah, and also the founders. Thank you.